Hello, I'm Dr. Kathuria, and welcome to our podcast, Conversations with an Organ Engineer. Here, we are spearheading breakthroughs in organ tissue engineering using induced pluripotent stem cells. Our purpose is to educate, engage, and inspire.
Hello everybody, welcome to our channel, Conversations with an Organ Engineer, where our organ engineer is Dr. Kathuria. And before we get into all the unpacking your whole journey, I do want to talk about some intros for us. So I would like to know who you are.
Hi, so I am Ananya, a graduate student studying Masters in Biotechnology at Johns Hopkins and working at Dr. Kathuria's lab since the past 6 months with stem cells.
And I am Alberto. I am an incoming second-year master's student here at Johns Hopkins University studying biomedical engineering. And I am also working with the wonderful Dr. Kathuria. Dr. Kathuria, when and why did you become a professor?
Okay, so my journey started with science when I was 16 or 17 years old, and I loved the idea of understanding the genes and the cells and how they function, how they work, and I never wanted to go to the corporate world. I always wanted to teach and engage and work with young minds. I'm a chatterbox, so I like to chat a lot. So this way teaching becomes fun. And so I always wanted to teach. So I started my journey at University of Minnesota, where I did a double major in neuroscience, and the second major was genetics, cell biology, and development. I was 17 years old when I moved to the States to just do science. And that's how I started. That got me into understanding the brain, the cells, as I mentioned. I used to work with those. And then did a master's in clinical neuroscience. Then I wanted to know how disease would affect our body. And how we can try to find cures for these diseases, understand how a little disease can, or not a little, a disease in general can affect the littlest of cells, and something goes wrong and then our body gets affected, we can't properly function. So I did the clinical aspect of neuroscience. And that led to my PhD in neuroscience, where I was trying to understand the intricacy of how autism develops in little children. And again, I was very focused on cellular development of autism. And that led to a postdoctoral fellowship at Harvard Medical School. So I was flip-flopping between US and UK.
Whoa.
So, as I said, undergrad was University of Minnesota, master's was clinical neuroscience, and PhD was at King's College London and Cambridge University. I spent 6 years there learning the skill of neuroscience and cellular biology, and came back to Harvard Medical School, where I was a postdoctoral researcher and a fellow for 2 years. And then I was promoted to junior faculty as an instructor of investigation there. And lastly, I was just promoted to assistant professor at Harvard Medical School, but I'd also wanted to get into the idea of engineering cells and understanding cells from an engineering perspective, tissue from an engineering perspective. And then I got the opportunity to come to Johns Hopkins School and Johns Hopkins Medical School. And now I'm an assistant professor here, where I teach students like yourself in the lab, in the classes. Biomedical engineering and neuroengineering is my specialty.
So to shift gears or get into more another topic, I want to talk about our name. So, what does Conversations with an Organ Engineer mean? What's our purpose for creating this podcast, Annie?
So the reason for creating this podcast is, as we said it in our intro, and you said it as well, is to educate, engage, and inspire. I want to inspire the next generation of cell biologists, engineers, and medical students to understand what the concept of tissue engineering is. And that's my specialty. So I do tissue engineering, and this brings me to the next topic that we will talk today about is what is tissue engineering and how do we make this organ engineering a reality? So do we ask questions now?
Yeah, I know you mentioned organ engineering, but how exactly do we get to that, I guess, that scale of things? You know, is it like a top-down type of thing, or do we start with minimal like cells and then work our way up?
So we start with the concept of stem cells. And the first question I would have as someone who is new to biology is, what are stem cells? So stem cell is a cell that has the ability to make itself into any part of the body. So the example I give during my lectures is, think of a kid and who wants to grow up into a doctor or an engineer. Or engineers here, possibly doctor here, or the idea is the stem cell has the ability to grow into a heart cell, a lung cell, a brain cell. It just requires a little bit of feeding in the right direction and directing us in the right direction.
And when you say any type of cell or create organs of any type, like stem cells can like be the eyes or like our kidney or or even
absolutely.
Our brain? Or?
Absolutely. It can these stem cells have the ability to generate into any cell type of the body. And our lab has generated various types of cells, like we've generated brain cells called neurons. We've generated liver cells, such as we call them hepatocytes. So we generate a lot of different types of the cells in the body.
Because this one cell is able to generate into different body parts, do you think that there are different kinds of stem cells that lead to forming these different organs?
So yes. So before I get into if stem cells have precursors or baby stem cells that turn into neurons or brain cell types, I would like to say the stem cell population or stem cell connotation in the scientific community is very broad. There are various kinds of stem cells. There is not one kind of stem cell. There is embryonic stem cell, there is induced pluripotent stem cells, there is mesenchymal stem cells, cord blood stem cells. The stem cell in itself is a very broad term. So I would say stem cells, there's a lot of variety of. The stem cells that we work with is induced pluripotent stem cells. And these are different and do not share as much as ethical concern as an embryonic stem cell would. So I want to distinguish in this particular first podcast what is an embryonic stem cell versus an induced pluripotent stem cell. An embryonic stem cell is a stem cell that is taken from a human embryo after 4 to 5 days of fertilization. And this has had a lot of ethical concerns for the scientific community as well as the broader audience of our our non-scientific community and general students, families, this concern is legitimate, and we're not unvalidating that concern. It is something that people are passionate about. But what we are doing is not embryonic stem cells. We are using induced pluripotent stem cells. And I want to talk about what induced pluripotent stem cells are. Induced pluripotent stem cells, I'm making it very, very clear, we're using those in our projects to generate these different cell types and body types we were talking about. Induced pluripotent stem cell is a technology that Yamanaka in his group in Japan won the Nobel Prize in 2012 for. What they did was they started with the mouse fibroblast, which means a mouse skin cell. And what you can do from that skin cell, so I can take my skin cell, and in their case, they took a mouse skin cell. So I take the skin of my hands, literally punch it, I get a skin cell. I can expand that skin cell in the laboratory in our tissue culture. Stay tuned, we will show you our tissue culture soon. And then we can expand those skin cells, and using genetic factors, we can reprogram them. What does that mean, reprogram them? Mm-hmm. Is that we can turn them back, go back in time and turn them into stem cells. So a skin cell can be converted into a stem cell.
And then that's where that induced term comes in, because you're saying you're reverting it back to a state that it once was.
Yes, we're taking it back to time. It doesn't require a human embryo. It doesn't require people to give their cells without consent. So, for example, I want to study my stem cells. What I would do is take, again, a punch of my skin, convert it into stem cells by reprogramming them. So I'm going to go back in time, convert it again into that stem cell state, and then use that. The advantage of doing this induction process that you mentioned is that it carries my genetic signature. Mm-hmm. So
it's personalized.
It's just personalized for me. So if I do Ananya's skin cells, it will be her genetic makeup. Mm-hmm. If I do yours, it will be yours. Mm-hmm. Alberto's genetic makeup. This gives us the advantage of looking at your cell types in real time. So that's what the concept of induced pluripotent stem cells is.
One question that I always had was, in our body, there are so many factors that contribute towards the working of our body as a holistic person. So when we take these skin cells and try to work with them outside our body, do you think what we are giving them manually translates to how they might work when they are part of our body? Does the microenvironment of our body get replicated at an accurate level when we try to work with cells outside?
My hope is yes. I would answer that question, no, we can't replicate the whole human body because our body is way more complicated and beautiful structure, which is a lot of us are trying to understand. But what we can do is replicate at least 50% of it. So I would say 50%, that's my opinion. And enough to make from your skin cells the reprogramming to stem cells into the cell I want. And that's the concept of tissue engineering or organ engineering. And so to explain it more further, for example, I took Ananya's stem cells here, or skin cells, reverted back them to a stem cell, and once we reverted back them into stem cells, we can if Ananya wanted to look, how does my brain function? We would convert that into brain-like cells called neurons, as I mentioned before, or we can actually now convert these into organ or 3D-like structures, so brain-like structures. And we have done this in the lab multiple times, that we've converted someone's skin cells into stem cells and then into 3D brain-like structures, where we can look at a brain organ or organoid-like organ-like structures. We call them organoids. The concept of brain organoids or mini brains comes from there.
When you talk about getting these skin cells and reverting back to a pluripotent state or a stem state, how exactly does that look like? Because I'm sure for the viewers, a majority of the population, I'm assuming, hasn't done any, I guess, tissue culture.
Yeah.
Or are we literally getting those cells and putting them in like a little plate, or how does that look like,
So, well, that's why you have professors like me to train you. But so students, I would put this message out, and anyone who's interested in science, not just students, anyone who's interested in science, the way we do this is when we do our reprogramming or reverting back to time for these cells, we actually have a step-by-step process. And this process I mentioned before was Yamanaka's group in Japan that published in 2007, and then 2008, first they used mouse, then they did humans cells, was converted, and they made a whole protocol for it, just like a recipe for making a cake.
Mm-hmm.
So it's not as easy as making a cake, and I would not recommend doing it at home because you don't have the proper facility. What you would do is you would need an infrastructure of a lab, specifically a biosafety level 2 lab, which you will find at universities or biotechnology companies or pharma companies and other kinds of laboratory grade settings. And then you just have this step-by-step 15 to 20 steps of making stem cells. But what you do, again, you take your skin punch, you give me the sample, I go into our sophisticated tissue culture areas that are sterile, clean, maintained, and we convert these skin cells back into stem cells, revert that. So we usually use plastic dishes, but we wear gowns, we wear gloves, we have our sterile equipment.
Like a whole setup.
A whole setup. And it takes about 3 to 4 months to reprogram these or induce them. And to get to the quality of stem cell that can then be differentiated or can be then engineered to be differentiated, that's where the organ engineering aspect comes into the different cell types.
I do want to talk a little bit just about how it feels to kind of grow these cells, right? Because before I came here to Johns Hopkins University, I've personally never worked with any like cell culture or any of those types of protocols. But the first time I did it, and I work with primary cells, so from rats, specifically rat brains. And the first time I cultured them, when I saw astrocytes, because I cultured astrocytes, a type of neural cell, it was just the most, to me, it's going to sound so cliche, but it was like an amazing thing, right? Because it's like you can't see these things, but under a microscope, they have these very distinct structures. And just to imagine that you could take that further and grow whole like 3D like
organ-like
structure, like to me that's amazing,
Yeah, whenever I talk to a broader audience, I usually get is this science fiction or real? And I would say it is, until you imagine it, it's science fiction.
Yeah.
That's why we have the audience, broader audience, to inspire us and to give us more ideas. So everything is science fiction until it's reality.
Yeah.
And I agree with you on, if you would have asked me 20 years ago if this was possible, I don't know.
Mm-hmm.
But now, when we're doing it in-house, we do it all the time. We've Ananya, how many brain organoids have you generated in the last 6 months? Brain organoids.
A lot of them. So now that we've generated so many brain organoids or 3D models of brain in our lab, for someone who's just starting out or trying to understand what exactly what 3D modeling is or what an organoid is, how would you explain the applications of these models in real world?
So just because I just said it was science fiction and it's kind of cool to look at these 3D brain or brain-like structures, what is the real-world application? Not just making brains in a dish is cool. It is super cool, as Alberto pointed out.
Cool.
And it is fun to play around with these cells. The real-world applications are multiple. One of the biggest things with especially the neuro world is any neurological disease like Alzheimer's, autism, Parkinson. We get to see the brain at the end stage when the patient has died. We don't know when the disease developed, schizophrenia or any other disease that involves the neurological aspect. We see the end stage. Once the person has already gotten it and is dying of it, and has no cure for these, that's when we see the brain. So the advantage, then people donate their brain and then we see, oh, that was what went wrong at the end stage. The advantage of it, I cannot ask Ananya to, okay, let's open her skull
Let's
and
cut
look
her
at it is not possible. And I can't do that with a patient either. So the advantage is I can take someone that has autism, schizophrenia, or Alzheimer's. I can take their patient's skin cells, convert them back to stem cells, convert them into these 3D brain-like structures, and track their development in real time. So I can see, as I said, the magic of the love for my science is for cells and understanding how cells develop. So I can see during real time when something goes wrong with these cells and what goes wrong with these cells. If I can understand in real time what goes wrong, I can find better drugs for it. So that is one of the biggest applications.
And I just want to add some input that kind of goes with the cells being personalized too.
Yes.
You know, these are very specific, patient specific. So with someone with bipolar disease, you can see how their brain works and how they develop, and hopefully implement drugs or protocols to I'm blanking, to impact that, you know.
We'll do an all new episode on personalized medicine and how it impacts regenerative medicine next time.
Mm-hmm.
But for now, I agree with you that since this is that person's genes and then we're deriving it, that this is very personalized and it is person specific, and this gives us a huge advantage on understanding why things go wrong for that particular person. This brings us to our conclusion for today. I hope you guys understood with our questions and QA, the Q&A answers by the students, and if you're a high school student, if you're just a mom with kids who wants to understand more and learn more about organs and organ engineering, I would encourage you to subscribe to our channel, follow us, DM us so that we can bring more content like this. And the way we're operating is we'll have an Instagram channel and a YouTube channel, and somewhere this is going to be on Spotify. We're still figuring that one out.
We'll see.
We will see. To be decided. But we would like us... we would like you to DM us all the questions you can think of, and meanwhile, engage with us as many questions. Meanwhile, I also encourage students who are interested in this topic. I help a lot of students learn about more science, the cool brains that we make, the cool kidneys that we'll make, or lungs, whichever organ you can think of. So DM us questions.
Leave a topic of an organ for us to discuss next time.
Yes,
we can
that's
discuss an eye, a brain, a stomach, anything.
Yeah.
And any final remarks, Miss Ananya? All right. Well, thank you guys for tuning in, and we'll see you guys when we see you. Yay!
Until next time.
Until next time. Yeah. See you when you see you. Hey everybody, it's Alberto here, and I just wanted to say some concluding remarks before the episode ends. We are on Spotify and we are on YouTube, currently trying to figure out the Instagram and the Apple Music podcast stuff. But for now, if you have any questions for Dr. Annie Kathuria or myself or anyone else on the podcast, make sure to send us an email of your questions or any comments of topics you want to discuss. Our email is at convos, CONVOS, organengineer @gmail.com. So it's convos, organengineer @gmail.com. Shoot us an email, anything you want us to talk about next time, and we'll make sure to get back at you guys. Yeah, hopefully you guys enjoyed the episode, and we'll see you guys next time.